Hi, everyone. Thanks for joining today. We will get started on the hour. Hello, everyone, and thank you for joining today. Welcome to the NOAA Omics Seminar Series. I'm your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from the series are made available on our YouTube and posted on our NOAA Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation, and at the end, I will read them for the presenter to answer. So with that, our presentation today is titled Gene Expression Responses of Stony Corals to Ocean Acidification from Shallow to Mesophotic Reefs by Dr. Federica Scucchia. Now I'll go ahead and allow Federica to share her slides. Dr. Federica Scucchia received her bachelor's in science in biological sciences from the University of Bologna, Italy, where she pursued her master's under a joint program between the University of Bologna and the University of California, Santa Barbara, where she focused on coral biology and gene expression. While at UCSB, she obtained her AAUS scientific diving certification, which allowed her to dive for her research on corals during her PhD at the Inter-University Institute for Marine Sciences in Eilat, Israel. There, she focused on the influences of environmental factors on the physiology, morphology, and gene expression of stony corals across life stages and depths. Federica joined the Martindale Lab at the Whitney Laboratory for Marine Bioscience, University of Florida, last year as a postdoctoral associate. Her research is focused on utilizing the sea anemone Nematostella as a universal expression platform to investigate various aspects of biomineralization for environmental restoration purposes, for example, coral reef conservation, using a variety of techniques, including transgenesis and protein engineering. Thank you, Federica, for presenting today, and I will now pass it off to you.
All right, thank you, Nicole, for the great introduction. Hi, everyone. It's such a pleasure to be presenting today for the NOAA Omics Seminar Series. My talk will be focused on some of the work that I've done during my PhD in Israel at the University of Haifa and in Eilat on the Red Sea. Particularly, I'll talk about the effects of ocean acidification on stony corals across depths, from shallow to mesophotic reefs, focusing on the gene expression changes that such reduction in oceanic pH can drive. So let's talk about corals. I'm sure that you're all already aware that in tropical and subtropical oceans, corals create reef frameworks that support highly productive and biologically diverse marine ecosystems. Among the major reef builders, we have scleractinian corals, also called hard or stony corals. These marine animals are mostly colonial, forming a multipolyp colony of clones that have a common calcium carbonate skeleton with different shapes depending on the species, the location, depth, the amount of water movement, and many other factors. But corals are not alone. They live in symbiosis with dinoflagellate algae of the Symbiodiniaceae family. These endosymbiotic algae reside inside the coral cells, this way getting protection from the environment and photosynthesis substrates, and supply the coral partner with the products of photosynthesis, in particular carbohydrates, which are crucial to sustain the energy demands of the coral metabolism, reproduction, and growth. During photosynthesis, the algal symbionts rely on light as the primary energy source, limiting the distribution of many coral species to the photic zone. However, corals are able to persist in deeper waters despite the limited light availability, and they can form reef structures referred to as mesophotic coral ecosystems, which can range in depth from 30 meters to almost 150 meters. Mesophotic coral ecosystems are deep reef communities consisting of diverse low light adapted scleractinian corals, microalgae, and sponge communities. The physical environment of these deep reef communities is characterized by strong gradients in downwelling solar irradiance, that is the predominant environmental abiotic factor, and by modest changes in seawater temperature and nutrient fluxes. Mesophotic coral ecosystems have been gaining a lot of interest since they appear to be generally protected from many of the local and global impacts that are affecting shallow water coral reefs, such as, for example, storm events, sedimentation, as well as increased sea surface temperature and associated bleaching. So for this reason, it has been hypothesized that mesophotic coral populations may serve as critical ecological refuges for coral species by providing a source of recruits to recolonize impacted shallow reefs and by offering shelter against disturbances and stressors. And this is known as the deep reef refugia hypothesis. As I have mentioned, light intensity and spectrum are the most significant environmental factors that differ across depth, and they represent important selective factors for corals due to the dependence on the photosynthetic activity of the algal and the symbionts. And because of the strong difference between the high light levels in shallow reefs and the low light levels in mesophotic reefs, some coral species have adapted to live in only one of these environments, so they are found only at shallow or only at mesophotic depths, while other species have adapted or acclimated to handle the range of conditions found in both environments. And such coral species are called depth generalists, and they are characterized by morphological, physiological, genetic, and symbiont-related differences that contribute to their persistence across depth gradients and light conditions. The deep reef refugia hypothesis has been highly discussed in the context of the increasing concentration of carbon dioxide in the atmosphere, which leads to the warmer sea surface temperatures and to ocean acidification, which is a global phenomenon caused by the absorption of such increasing CO2 by seawater that through a series of chemical reactions leads to the reduction of the oceanic pH and of the concentration of carbonate ions in seawater. Ocean acidification is one of the major threats to the future of coral reefs, as declining seawater pH has been shown to severely reduce the capacity of stony corals to grow their skeleton, and predictions suggest that the structural integrity of most reefs worldwide will be negatively impacted due to the reduction in coral skeletal density. However, studies also show that coral response to simulated future ocean acidification conditions is highly variable, and this increases the uncertainty about the extent of the ocean acidification threat to coral persistence, since both negative responses and signs of potential acclimations have been reported. In addition, such uncertainty is partly due to the ability of corals to survive in naturally acidic environments, such as near volcanic seeps, for example, which sheds light on the potential for coral acclimation and or adaptation to the projected acidification scenarios. So far, all studies on the effects of ocean acidification have focused primarily on corals inhabiting shallow reef systems. For example, there is a study conducted last year, a couple of years ago, where Cornwall and co-authors provide quantitative projections of how ocean acidification will influence the carbonate production across coral reefs in all major ocean basins, incorporating the results of more than 100 studies across the globe, which were all based exclusively on shallow corals. So if we go back to the hypothesis that mesophotic coral ecosystems represent a potential source of recruits to replenish deteriorated reef zones, a lack of understanding of how mesophotic corals will respond to the predicted ocean acidification conditions restricts our projections of how of the refugia potential of these ecosystems and of how the species composition of coral reefs across shallow to mesophotic depths will change in the future. And so during my PhD, I worked on assessing the genetic and physiological responses to future ocean acidification conditions of an abundant Indo-Pacific stony coral species, Stylophora pistillata, which is a depth generalist coral species that inhabits shallow and mesophotic depths. And this was done in collaboration with Professor Ollie Putnam at the University of Rhode Island. What we did here is collecting fragments of adult Stylophora pistillata colonies from 5 meters and 45 meters depth in Eilat on the Red Sea, and we exposed them under simulated future ocean acidification conditions in a controlled environment aquarium system at the University of Haifa. Specifically, we manipulated the carbonate chemistry of the water by injecting CO2 to reduce the control pH 8.2 and obtain the target values of pH 7.8 and 7.6, which are the predicted future mid-century and end of the century seawater pH conditions according to the IPCC business as usual emission scenario. And at the end of the incubation under acidic conditions, after a bit more than a week, we collected the fragments and we explored changes in gene expression, as well as in photosynthesis and physiology between shallow and mesophotic individuals. So before I jump into the results, I'd like to give a more detailed overview of the RNA-seq analysis that we performed. So we sequenced the total RNA that we extracted from fragments collected per each depth and pH conditions, and we obtained about 23 million reads per sample. We then fed the raw reads into an RNA-seq processing pipeline, starting with cleaning and filtering the reads, blasting them against available proteome databases of Symbiont species to identify the algal symbiont species composition at each depth. And then we mapped the clean reads to the genome of Stylophora, which is available on NCBI. And then we proceeded with the differential expression analysis and the gene ontology enrichment analysis. We also conducted a single nucleotide polymorphism analysis to assess the genetic differentiation across depth by essentially using an identity by state analysis, which basically computes the pairwise fraction of identical alleles between samples. So getting to the results, overall, we found that mesophotic corals possess a greater capacity to cope with the effects of ocean acidification compared to their shallow counterparts. But let's see why. So first, we saw that the metabolic rate of shallow corals is negatively impacted by acidification, while mesophotic corals appear to be unaffected by exposure to acidification conditions. It is known that acid-base parameters such as pH and CO2 are considered to affect metabolic processes and to induce changes in the ion exchange transport system that regulates intracellular pH. And looking at our RNA-seq analysis, particularly at the results of the gene ontology enrichment analysis, we found that mesophotic corals exhibited a larger activity of cellular functions linked to acid-base regulation compared to shallow corals. The graph here shows enrichment of these processes in mesophotic corals compared to shallow corals at the lowest pH. And this suggests that mesophotic corals possess a greater capacity to adjust to the drop in pH, counteracting the cellular effects of seawater acidification. And this potentially explains the maintenance of unaltered metabolic rates. It's worth mentioning here that a few works that have explored the gene expression changes in corals living in naturally acidic environments have found that the persistence of such coral communities in low pH waters appears to be supported by the differential expression of pH homeostasis genes, so similar to what we see here for mesophotic corals. And we have also observed that mesophotic corals possess a higher baseline activity of cellular functions linked to cell-to-cell adhesion and extracellular matrix compared to shallow corals. But let me navigate you through the graph first. In the upset plot on the left, I'm showing all the shared gene ontology terms that were enriched in all possible comparisons, so among all depths and pH conditions. And when we look at the intersection among all comparisons, so at the terms that were enriched in all cases, we can see a subset of gene ontology terms shown in the heat map with the percent of differential expressed genes that were upregulated and downregulated in each term across pH conditions in mesophotic corals as compared to shallow corals. And so here we see that cell-to-cell adhesion and extracellular matrix genes were upregulated in mesophotic corals as compared to shallow corals at the control pH, indicating that mesophotic corals have a higher baseline expression of these functions under normal pH conditions. And this could be a critical asset enabling tolerance to acidification conditions. In fact, it is known that cellular pH can profoundly alter processes such as cytoskeletal integrity, cell-to-cell adhesion, and cell coupling. And cell-to-cell adhesion is the binding between cells and the extracellular matrix. So having a higher baseline activity of these functions contributes to preserve physiological cell-to-cell adhesion properties under acidic pH. The reason why mesophotic corals have a higher baseline expression of such functions remains to be determined, although we suggest that this derives from the fact that as light gradually decreases from shallow to mesophotic reefs, the decrease in photosynthesis rates with depth leads to increasing levels of CO2 and consequently to acidosis. And in mesophotic corals, evolution or adaptation or local adaptation might have favored the constitutive higher expression of specific genes, such as cell adhesion and extracellular matrix genes, to deal with lower levels of intracellular pH. There are also other aspects that contribute to the capacity of mesophotic corals to maintain their physiological integrity under acidification, with one being a greater use of lipid stores, which ensure an efficient mobilization of energy reserves to meet physiological demands under stressful conditions. Also, if we look at the tissue biomass, so the living tissue covering the skeleton, we see that mesophotic corals have a much lower tissue covering their skeletons. And this is a characteristic that in stony corals has been shown to promote a more energetically efficient proton removal out of the epidermis, which ultimately promotes calcification and the maintenance of physiological homeostasis. The coral capacity to withstand future ocean acidification conditions is not only determined by the coral host, but also the algal partners can play a critical role. When we look at the enriched functions in mesophotic corals across pH conditions, we observe an enrichment of functional pathways linked to carbohydrate binding and transport. And the main source of sugar for corals is the endosymbiotic algae with their photosynthetic activity. So based on these results, it seems that the increase of CO2 in seawater, which is what lowers the pH, stimulates the photosynthetic activity and efficiency of the symbiotic algae living inside the coral, which we indeed found when measuring the photochemical efficiency of the algae using pulse amplitude modulation fluorometry. Sugars represent a major energy source for corals, and a greater photochemical efficiency and an increased translocation of photosynthesis products from the symbionts under ocean acidification has been indicated to be critical for the ability of corals to withstand the effects of ocean acidification by increasing the energy available to the host and the metabolic supply of carbon to the calcifying fluid, both of which support calcification. And if we look at the photochemical efficiency data, we see that the endosymbionts photosynthesis is stimulated in both shallow and mesophotic corals, but we only found that an enrichment of sugar-related metabolism in mesophotic corals, which, based on what I've shown you so far, are the ones that were able to maintain their physiological integrity under acidification. So the photochemical response is similar across depths, but the utilization of the photosynthesis products is not the same. And in this regard, we have to consider that shallow and mesophotic Stylophora pistillata corals host different symbiont species, which we identified by blasting the RNA-seq reads against all available proteome databases of Symbiodiniaceae species. In particular, we found the shallow coral host Symbiodinium adriaticum, macroadriaticum, and mesophotic corals host Cladocopium boroi, which are known to possess broad physiological differences. And this indicates that it is the specific host symbiont partnership that determines the response as a whole to change in the environment. Also, I think it is worth mentioning here that, however, some coral species that are adapted to naturally high PCO2 environment have been shown to mainly harbor symbionts of the genus Cladocopium, suggesting that Cladocopium species may indeed possess a better capacity to thrive under high PCO2 conditions, so under low pH conditions. So if we put all of these results in the context of the deep reef refuge hypothesis, something else that we need to assess is the degree of connectivity along the depth gradient, which would indicate whether there is gene flow between shallow and mesophotic corals. Here we estimated the genetic diversity from transcriptome-derived single nucleotide polymorphisms, and using the principal component analysis based on the pairwise fraction of identical alleles, we found a strong clustering by depth, indicating that shallow mesophotic corals are not genetically connected. And so that no gene flow through larval disruptors occurs for this species at this location. So this means that within the Gulf of Eilat, mesophotic Stylophora pistillata corals have a low potential to reseed and repopulate shallow reefs. However, these results correspond to the local trend in the Red Sea and may only be specific to Stylophora pistillata, since, for example, in another study, we found genetic connectivity among shallow and mesophotic corals in Bermuda for the coral Poraites asteroides. And also other studies have found connectivity for other species at different locations across the globe. And so if the ocean acidification tolerance traits that we detected here are shared with other mesophotic coral species, in those locations where gene flow between depths exists, shallow corals may obtain stress-tolerant larvae and therefore traits from mesophotic conspecifics. And so this would gradually build the reef resilience through adaptive evolution. So to sum up, using a combination of RNA-seq and physiological analysis of the coral holobiont, we have found that mesophotic Stylophora pistillata corals possess a greater capacity to cope with the effects of ocean acidification compared to the shallow counterparts. And we have seen that such ability stems from both host genetics and physiology and from symbiont-based differences. Specifically, we propose that this capacity is derived from the combination of having a higher expression of genes related to the maintenance of cellular homeostasis, such as cell-to-cell adhesion and extracellular matrix genes, but also genes related to the regulation of cellular acid-base parameters. And this is combined to physiological characteristics, such as the distribution of living tissue in terms of thickness that can improve ion exchanges, and to symbiont-related processes, such as increased photochemical efficiency that may lead to a greater translocation of photosynthesis products to the coral host, as suggested by the enrichment analysis, which would support the maintenance of physiological integrity. We have also seen that shallow and mesophotic Stylophora pistillata corals at this location are genetically differentiated, suggesting that there are environmental barriers to gene flow between depths. So that there is low potential for mesophotic corals to provide recruits to repopulate degraded shallow reefs. However, in other locations, corals are found to be genetically connected, and if these traits observed here are shared with other mesophotic coral species, shallow corals may obtain stress-tolerant recruits from their mesophotic conspecifics, which would support the refugia hypothesis. And so to conclude, I would like to thank the coral team, particularly all of my former lab members and my PhD advisor, Talimas, as well as our collaborator, Professor Putnam, and all our funding sources. And finally, thank you all for joining the talk today and for listening.
Thanks, Federica, for this very interesting talk today. We really appreciate you volunteering for the seminar series today. We'll move on now to take a few questions from attendees, and then conclude. So no questions yet, but I do have a question for you, Federica.
Yes.
On your talk. So, and I think this is really exciting research, as actually NOAA Ocean Exploration has explored the Blake Plateau and one of the largest deep sea coral reefs known on the Blake Plateau. This is news that was released earlier in 2024. And my question for you is, with what was exciting about this, the exploration of this plateau was they had discovered a large deep sea coral reef, almost the size of Vermont. And I'm wondering if it would be possible, or on your perspective on corals with increasing resistance with depth. And if you would think or propose that corals at deeper depths, over 150 meters, maybe over 200 meters, you would see the same resistance or gene expression.
Yeah, so that's a very interesting question. There are a few other studies that have explored both in terms of gene expression and physiology or even morphology, corals inhabiting such deeper reefs. And there's also, like, for example, the cold water corals, the ones that don't have symbionts, that seems to be resistant to some of the stressors, but also they are protected from some of the stressors, so they don't have the same increase of temperature of shallow reefs. So it will be very interesting to see if other corals at other locations, even deeper than these studies, so even deeper than 45 meters, have the same characteristics. So something that we don't really know, for example, in these experiments, we have used the same pH decrease for all corals, but we don't really know if all mesophotic reefs or deeper than that will experience the same decrease in pH, for example, in the future. So it would be also, I think, interesting to kind of monitor the pH and the temperature change across the water column, which is also based on the local pattern of upwelling and currents.
Thanks for that. We did have one question come in. This one's from Allison, who says, great talk, really enjoyed it. Can you explain why you blasted sequences specifically with the Symbio database and not with a general blast? Is that in order to separate out genes specific to Symbios and the host?
So yeah, so we wanted to, it was done mostly to, because corals host different symbiont species, they have a community. So we really wanted to look at the most abundant one. So that's why we use the blast to kind of find the most abundant species. So with the blast, you see the majority, where the majority of your reeds goes. So only the symbiont related reeds were, the host-related reeds were not basically aligning to any of the symbiont related proteomes, so we wanted to only see the algae one. But then when we mapped to the coral genome, we didn't really care about those reeds because those were not mapped to the genome of the coral, which this one on NCBI is only corals gene. So the reason was just to find the abundant algal species per each depth, which is also affecting then the response of the coral to the change in the environment.
Thanks, Federica. Our next question comes from Jeff, who says, thanks for the great talk. Sorry if I missed it, but is there, or if there, Jeff was asking if there might have been changes in mesophotic coral growth with the increased photochemical efficiency.
So yeah, that's a great point. So the length of the experiment was quite short. So we wanted to do like a very short-term experiment to see the responsiveness among the two corals. So with that timeline, I wouldn't expect any growth change, because for corals, it takes a lot more to see any change. And generally, mesophotic corals have lower growth rates than shallow corals. So I would exclude that. But for sure, for in a longer, long-term experiments, yeah, you will see changes in growth, which you can monitor by, taking pieces of the skeleton and just by measuring the mass change, for example, over time.
Thank you. Thanks, Federica. That looks like we've come to end our questions. So we'll give it one more minute before we conclude today. Well, with that, thank you so much all for attending today. Thank you, Dr. Scucchia, for your great presentation on deep sea or on mesophotic corals and gene expression in relation to ocean acidification. And with that, thanks everybody for joining. Our next seminar will be on the third Wednesday of June at 2 p.m. and details will be available shortly via our listserv. Definitely check out the NOAA Omics website and landing page if you have not yet already. There's a link in the chat. And thanks Federica for joining today.
Thank you so much.
Bye everyone, see you next time.